Hi, my name is Lisa Barcelos, and I am a professor of epidemiology and biostatistics in the School of Public Health at UC Berkeley. I'm co-leading this research study on COVID-19 in East Bay Area communities with my colleague Eva Harris. We recently invited one adult from more than 350,000 households in 12 cities to participate in the initial screening phase. Now, in phase 2, we have randomly selected 5,500 individuals for testing and additional data collection. This will help us determine the prevalence of both current and past coronavirus infections in the East Bay and to monitor changes in the spread of infection over the next 6 to 8 months. Please, if you can, take the time to watch the rest of this instructional video. We have put it together to help guide you through the oral, nasal, and blood sample collection that's needed to do this study. We want to thank you for participating and for making this important research possible. Please don't hesitate to contact us at COVID-19survey@berkeley.edu or by phone at 510-643-0580 if you have any concerns or questions. Thank you very much.
Hello, my name is Eva Harris, and I'm a professor of infectious diseases in the School of Public Health at UC Berkeley. And I am co-leading this study of COVID-19 in the East Bay with my colleague Lisa Barcelos. We think it's an important study for understanding risk factors for infection and, importantly, understanding safe ways to go back to our lives as we reopen society. And I just want to add my thanks to Lisa's and thank you very, very much for participating in this important study with us.
Hello. Thank you for participating in our Bay Area coronavirus study. Detailed instructions can be found within the sample collection kit. If you have any questions or concerns, please email us at covid19survey@berkeley.edu or call us at 510-643-0580. Sending an email to covid19survey@berkeley.edu will give you the fastest response. You can also call 510-643-0580 and leave a voicemail. We will return your call as soon as possible. There's only very minimal discomfort from the application of the lancet for blood collection, but it may be easier to have someone assist you. The swabs collection kit contains the 2-inch binder clip, the swab Ziploc bag containing absorbance paper, 2 swabs, the paper ruler, the collection tube containing reagent, parafilm, an alcohol wipe, and bubble wrap and a rubber band. The blood collection kit contains the filter paper in the Ziploc bag, the plastic tube with orange cap for the used lancet, gauze, bandages, 2 small lancets, 2 large lancets, alcohol wipes, and a large tissue. There is also a blood spot picture card included in your kit. The return box contains the original shipping box to be reused, the biohazard bag, the sample info card to record the date and time of each sample collection, the return label, the clinical pack bag, and packing tape. Reminder, please keep the box and be careful not to damage it so it can be reused to return your samples. For a FedEx pack return, these are your options. Give it to a FedEx truck driver, deposit it in a FedEx letterbox, drop off at Kinko's, or drop off at a FedEx store. Do not use Walgreens to return your kit. Please return your kit within 48 hours of collecting your samples. This is very important. You will no longer be collecting a saliva sample. Nasal swab and blood spot collections can be done at any time of day. Nasal swab collection takes about 10 minutes. We recommend eating, drinking, and moving prior to completing the blood spot. Please note, though the finger prick and blood spot take about 10 minutes, it requires 2 hours to dry, so please plan accordingly. Option: while waiting for your blood spot to dry, complete the online study questionnaire, which takes about 20 minutes. The link to this questionnaire is in your shipment confirmation email. Boxing up the contents of your kit will take about 10 minutes. Warning: the collection tube contains storage solution for your nasal swab. Do not touch or ingest. Wash with water for 15 minutes if solution comes into contact with your eyes or skin. Use the provided spill kit. Do not use bleach. Use the binder clip to place the tube upright during the sample collection. Retrieve the swabs from the swab Ziploc bag. Open the wrapper and without touching the swab tip, remove one swab. There is one extra swab in case it is needed. If not needed, you can discard. Gently insert the swab less than 1 inch, about 2 centimeters, into the nostril until resistance is met. Do not push too far. Rotate the swab gently several times against nasal wall. As you pull it out, rotate the swab around the inside of the nostril nare, which is where your nostrils flare. Hold it in place 10 to 15 seconds to absorb nasal secretions, and then repeat in the other nostril using the same swab. Align the ruler with the end of the swab. Break off the swab handle by bending it at the end of the ruler. Discard the handle. Place the swab into the collection tube, submerging the tip in the solution. Warning: do not touch the liquid in the tube. If the liquid spills, clean up with water and the provided spill kit and wash your hands. Do not use bleach. Cap and secure collection tube tightly, then invert tubes several times to mix. Wipe the outside of the tube with an alcohol wipe. Peel the backing off the piece of parafilm. Gently stretch the piece of parafilm, then stretch it around the tube cap tightly to prevent leakage. Wrap the tube in bubble wrap and secure with the rubber band. Then place the tube inside the swab Ziploc bag containing the absorbance paper. Then place the swab's Ziploc bag into the biohazard bag. Record the date and time of your nasal swab collection on the info card. If a spill occurs, please put on the gloves provided in the spill kit. Place a piece of dry paper towel over the spill to soak up all of the solution. Then wipe down the area with several fresh paper towels twice with fresh water. Do not use bleach. Only use water to wash the area. Dry area with fresh paper towel. Remove the gloves, then bag all the contents of spill cleanup and dispose in trash. We encourage you to eat, drink, and move around prior to the collection for best results. Remove contents from the blood spot bag. Unfold the large tissue and place it on a clean level tabletop. Without touching the discs, pick up the blood filter paper. Do not discard the BS filter bag. This will be used later. Draw an arrow pointing to the first disc you will apply blood to. Choose the correct lancet. We suggest participants start with one of the small white and blue lancets. If these do not work, 2 larger green and orange lancets are included. Choose either your ring or middle finger to prick, then wipe it with the alcohol wipe. Twist off the cap of the lancet and pull it straight out. Place the lancet firmly on your chosen fingertip. Press it into the fingertip, then press the top button to puncture. Apply pressure near the puncture site to obtain a blood droplet. It helps to stand up and drop your arm downwards to move blood toward your fingertip. If you need to use the larger lancet, wipe your chosen finger with an alcohol wipe. Twist off the green protective cap and pull it straight out. Press the lancet body firmly against the puncture site to activate the device. Gently apply intermittent pressure near the puncture site to obtain the required blood volume. Avoid touching the discs as you carefully pick up the blood filter paper. Apply hanging blood drops to each disc until the disc is completely saturated on the front and the back. Completely saturate 4, but preferably all 6 discs. Saturate each of the discs so that the front and the back contain blood. See the blood spot picture card in your kit for an example. Then place the blood spot filter paper onto the tissue. Wipe the finger with gauze and apply a bandage. Dispose of the used gauze and wipes. Record the date and time of completed blood collection on the info card. Let the blood filter paper air dry on top of the tissue for 2 hours. Do not let it touch other surfaces. Place the used lancet into the large plastic tube with an orange cap. Place this tube and the info card into the biohazard bag. After fully air-dried, put the blood filter paper into the provided BS filter Ziploc bag. Remove the desiccant from the blood spot bag, which is a small white pouch. Place this pouch into the BS filter Ziploc bag with the blood spot. Seal the bag with the filter paper and pouch inside. Wipe the outside of the bag with a fresh alcohol wipe. With the blood filter paper inside, place the BS filter Ziploc bag into the biohazard bag. Wash your hands when you are finished. Note: please return your kit within 48 hours of collecting your samples. Please complete the online questionnaire before returning your kit. There is a link to the questionnaire in your shipment confirmation email. This questionnaire should be completed on the same day you collect your samples. Press out any air from the biohazard bag containing your samples and used materials, then seal it. Place the biohazard bag in the original shipping box. Seal the box securely with packing tape included in the kit. Remove the original FedEx label from the box by peeling it off completely or defacing it with a Sharpie, including the barcode. Please be sure to deface the original label on your box to protect your privacy. Place the boxed kit inside the clinical pack bag. Seal the bag by removing the strip attached to the opening of the bag and secure the adhesive to the exterior of the bag. Place the return shipping label on the clinical pack bag in the designated space. For a FedEx pack return, these are your options. Give it to a FedEx truck driver, deposit it in a FedEx letterbox, drop off at Kinko's, or drop off at a FedEx store. Do not use Walgreens to return your kit. Thank you for taking part in our study.